Hello, everyone, and welcome to today's webinar introducing the latest version of the StarDrop drug discovery software, StarDrop 7.5. My name is Catherine, and I am delighted to welcome you to this webinar today. So with that, it's my pleasure to introduce you to today's webinar presenters. I'm delighted to be joined by our Chief Scientific Officer, Ed, Product Manager Ray, and Principal Scientist Mario. Ed has a degree in mathematics and started his career at Glaxo Wellcome, working as part of a pioneering team building predictive pharmaceutical tools. He developed the first graphical user interfaces for working with predictive models, which were adopted globally within Glaxo Wellcome. He was a core member of the team which established the UK operation of Comitro in 2001 and remained with that company, now operating within BioFocus DPI following merger and acquisition until 2008. During this time, he designed and built the StarDrop software and in 2009 co-founded Optibrium. Ray is responsible for product management at Optibrium, a role in which she applies her considerable expertise in software solutions development and computer-aided drug discovery, gained in technical and commercial management roles at CCG, ACD Labs, and Crescit. Ray has a background in theoretical chemistry, gaining her PhD from the University of Missouri, and since joining Optibrium in 2020, she has overseen multiple successful StarDrop and Sorella releases. Finally, Mario, our principal scientist. Mario has a background in computational chemistry with a PhD in natural sciences from Tallinn University of Technology, where he has also held roles as a lecturer and a research assistant. Since joining Optibrium in 2017, Mario has led much of the company's research and development efforts into metabolism prediction, developing new models based on quantum mechanics and machine learning for predictive modeling. Ed, Ray, and Mario, alongside a whole host of talented scientists and engineers, have been heavily involved in crafting StarDrop 7.5 and are really excited to share the latest updates with you. So without further ado, I will hand over to Ed to kick things off for us.
Thank you very much, Catherine, and welcome everyone to today's webinar. As Catherine said, we're really going to focus today on giving you an introduction to our latest exciting release of StarDrop 7.5. I'm aware that one or two people in our audience today aren't so familiar with StarDrop, so by way of an overview of what I'm going to do to set the scene, I'm going to kick off with a short introduction, just a short overview of what StarDrop's all about, before focusing really the majority of today's time on introducing all the new features that we've added in this latest release. I'm going to do that with a few slides to set the scene, and Mario and Ray are going to help me do that. And then we're going to dive into a live demonstration, just to give a feel for the software and actually using it before wrapping up with a bit more information. And during that last session, we'll obviously address any questions that you care to submit. So as Catherine said, if there are any questions that come up during today's presentation, please tap that button and drop them in, and we'll certainly get to them when we wrap up towards the end. So in terms of introducing StarDrop, as many of you are very well aware, StarDrop is a complete package of integrated software for small molecule design, optimization, and data analysis. It really embodies all of the research that we do in-house, all the cutting-edge science that we develop within an interactive and intuitive user interface. We developed StarDrop in a modular way. Much of what we put in there is developed in-house, but we also work with leading partners where we collaborate to bring in their technology. So there's a seamless way to use all of the StarDrop functionality and that collaboration, or the results of that collaboration in one place. We are, of course, aware that StarDrop is not used in isolation in any of your organizations. It needs to sort of work seamlessly with your laboratory systems, your databases. So it's designed to be flexible and easy to integrate with your own platforms and customizable to fit with your own workflows. And of course, StarDrop already has a large and global customer base, which includes many of the top 10 pharma, a whole range of different sized biotechs, academic groups, not-for-profit groups, and so on. In terms of StarDrop's core features, it's really focused on sort of taking it all the way through from that sort of initial selection process through to the design of individual compounds. At the heart of StarDrop is our approach for multi-parameter optimization called probabilistic scoring. So this enables you to define the profile of properties which are important for your projects so that you can quickly target those high quality compounds. Of course, it's not just the quality of compounds you need to focus on, particularly early on in a project. You also want to think about how to balance that quality against diversity. And so we have the ability to sort of consider the chemical space of all the molecules that you're considering. We provide ways to enable you to make selections within that, particularly with a view to helping you to avoid missed opportunities. Chemical space is just one of a number of different data visualizations that are available, all of which are designed to make it easy for you to understand the structure-activity relationships in your chemical series, whether that's doing specific SAR analyses or using StarDrop's unique card view approach for sort of really bringing your data sets to life so that you can lay out your compounds and consider the chemistry the way you want to think about it. And all of this is wrapped up in a sort of an interactive design environment so that you can use features such as StarDrop's glowing molecule that helps you understand which parts of a molecule are having a particular impact upon particular properties, helping guide the design of the balanced compounds. And of course, outside the core application, we then have a very comprehensive range of across a number of computational chemistry capabilities. So here I've got sort of 10 of the modules that we've that are available with StarDrop. In the top row, the first three, Nova, Biostar, Inspira, are all for sort of that compound generation sort of process. So Nova provides a number of techniques for enabling you to generate and prioritize new compound ideas. And Inspira takes it to another level by learning from your feedback about the ideas that are generated in order to then generate further suggestions built upon your preferences. And a lot of that is underpinned by the Biostar database, which has provides access to a large number of precedented transformations from the literature. We have a couple of modules which sort of focus on 3D space, CSAR, sort of more in the structure-based design space, enabling you to look at and explore docking results, and eSIM 3D, which is very much about ligand-based design. And Ray's going to tell you a little bit more about this as she introduces one of the sort of the headline new features that we've got within StarDrop 7.5 around virtual screening. The MPO Explorer module enables you to develop multi-parameter optimization strategies. And then we have a series of modules for providing predictions of different properties that could be important to your project. So the ADME QSAR module gives you high quality predictive models of key ADME properties. The Derek Nexus module provides knowledge-based prediction of toxicity endpoints. And the Metabolism module is one of the new features added within StarDrop, sort of replacing our original P450 module. And Mario's going to be telling you a bit about that in a few minutes. And we also have the Auto Model module. So this enables you to build and validate models based around your own chemistry and data and use them within your design processes. And finally, just a sort of a few words on flexible integration and customization. StarDrop's very much about trying to enable you to work with all of your data to get the most out of your research and your infrastructure. And that flexibility I described earlier is all about enabling you to tie StarDrop seamlessly to your other platforms and in-house tools. A couple of examples of this are the generic database querying interface to give you seamless access to your latest data, or the post-generation interface, which enables you to run docking and alignment calculations directly from StarDrop and retrieve the results and explore them directly within StarDrop to get that seamless experience of working with these different calculations. And of course, there are many different ways that you can link StarDrop with other third-party platforms, whether that's database platforms such as CDD Vault, Science Cloud, or Satara D360, pipelining platforms such as KNIME or Pipeline Pilot, vendor catalogs of molecules such as Molport, Enamine, eMolecules. And many of these, we have scripts available already on our community site that you can download and use so that you can interact with these and work with these platforms directly. But if there are any further examples that we don't have there, the whole environment is set up to make it possible to customize either by yourself or working with us. So if you have any suggestions for ways that you would like StarDrop to integrate with your platforms, we're always very open to discussing the possibilities with you. So I just wanted to set the scene with those few slides, as I said, for those of you less familiar with StarDrop, but now I really want to focus on what's new in StarDrop 7.5. And what we're going to do is start by focusing on the new Metabolism module, So it's at this point that I'm going to hand over to Mario to describe what we've done here.
Thank you, Ed. So I am going to talk about the new metabolism module. And I guess you've already realized that this has evolved from our old P450 metabolism module. And I thought I will give you a short overview of the capabilities of the old P450 module, and then we'll move on to the new capabilities of the new metabolism module. So the P450 module was or will be used to predict metabolic routes, sites, products, and lability of P450 metabolism. And we have 7 reach selectivity models for major human P450 isoforms, which output likely metabolites for you to analyze. Furthermore, we have the P450 model, which predicts the major metabolizing isoforms for a compound. And last but not least, one of the important models we have is the site lability model, which indicates the vulnerability to metabolism. So you could redesign your molecules if necessary. And here on the right, we have an example of how the P450 metabolism tab looked like. And when you have a specific compound, then you would usually start with the P450 model, which predicts the major metabolizing isoform. And for this compound, the 2 isoforms which are relevant are the P450 2D6 and 2C19 isoforms, noted here by that pie chart. However, the reach selectivity of those 2 isoforms is not the same. As you can see, the reach selectivity model predicts that the primary metabolite for 2D6 is this carbon over here to the right hand, called C9. And for 2C19, the primary site of metabolism will be the carbon denoted as C1. And last but not least, we also have a histogram which tells you the site lability. So if you want to know if any of those carbons are labile to P450 metabolism, then in this case, it would be the carbon C1. So if you think, or if you see that this metabolism would be bad in any way, then this is something, or this carbon would be the carbon which you would have to protect. Now, the new capabilities of the new metabolism module would be new enzyme families and new isoform-specific models. We have extended our models beyond human P450s, and we have added aldehyde oxidases, flavin-containing monoxygenases, uridine diphosphate glucuronyl transferases, and sulfotransferases. And as it was with the P450 models, these models are isoform specific. Furthermore, we have expanded the range of P450 metabolism models to preclinical species. And these models are very similar to the existing P450 models in the sense that they are based on mechanistic principles. And here on the right, you can see how we use quantum mechanical simulations to estimate the reactivities for the potential sites of metabolism. This example, what you just saw, was for the flavin-containing monoxygenases. And we take the reactivities, which we calculate using quantum mechanical approaches, and combine them with steric and orientation effects, which raise from specific isoforms, and combine them using machine learning approaches. Now, these models are rigorously validated by us using external test sets. However, we have also published them in peer-reviewed journals. So if you're interested how we did it, you can go and read about them in these specific journals, which are referenced down here below. Now we have added models for important isoforms for the aforementioned enzyme families. And as you can see, we've used different statistics to evaluate how good those models are. But I think the easiest statistics to look at is the balanced accuracy. And you can see that for each model, for those isoforms, the balanced accuracy is greater than 80%. Now, before we go to the questions which you could answer with the new metabolism model, I would also like to say that since we've gathered a lot of data for a lot of different enzyme families, then we also managed to put together another witch model, the witch enzyme model, and you can see an example of the output down here. So what you can do with the witch enzyme model is you can predict which enzyme family is the most likely enzyme family to metabolize your specific compound. And in addition to the witch enzyme model, we have also used the data available to us to train or find out rules or heuristics how to predict the metabolic pathways. And here on the right, you can see an example metabolic pathway where in the center we have the parent compound. And we have calculated 2 generations of metabolites for this parent compound. The first circle here represents the first generation, and the outer circle represents the second generation. And you would be probably interested in the metabolic pathway if you're not sure what enzyme families are relevant for your compound. These heuristics run the models, sort out the relevant metabolites, and present this in this nice graph. And you can see the cards which have a brown or yellow background are the ones which are experimentally observed as well. So the questions which you could answer with the metabolism module. First of all, which enzymes are responsible for my compound's metabolism? And you could use the which enzyme model to answer this. And you could also use the which P450 model to answer which isoforms are responsible for the metabolism of your compound. Also, you might observe that your compound is metabolically unstable. So these models will help you redesign your compound to improve the metabolic stability, and you can run your improved compound through the models again to see if the improvements you made have actually improved the metabolic stability. Then you can also ask the question, how can I reduce the risk of drug-drug interactions? And with these models, you can see if the compound you have has one or multiple routes of clearance. Either maybe your compound gets metabolized by 1 or 2 families, and if it does get metabolized only by 1 enzyme family, then maybe at least it gets metabolized by 2 isoforms, as we saw in the previous example 2 slides ago. Then the metabolism models will help you to understand which metabolites will be formed from your compound, and this is most likely the job for the heuristics or the metabolic pathway, which I talked about a second ago. And also, if you go through the results, you should be able to understand, are any of the metabolites which are predicted likely to be active or reactive or maybe toxic? So you know that if they are toxic, then you can redesign them so they would be more stable and less toxic. And last but not least, I also told you that we have expanded the range of P450 models to different species. So if you compare the results from human P450 models with preclinical species P450 models, these models should help you to choose the preclinical species for your toxicity studies. And I think that concludes the new capabilities of the metabolism module. And now I would like to give the microphone over to my colleague, Ray. Thank you.
Thank you, Mario. So I'm going to tell you a little bit about virtual screening using Surflex eSIM 3D within StarDrop. So like Metabolism, Surflex eSIM 3D as a module was integrated in StarDrop back in StarDrop 7.1, where we enabled a ligand-based design approach that would sit next to our CSAR tab, which gives us the structure-based drug design approach. And what this ligand-based approach does is it gives us the ability to design and compare ligands from the ligand perspective, as opposed to thinking about how it might interact with a protein. The value in this is that you can make comparisons between ligands without having any structural information available to you. So this is fully integrated within StarDrop and based on the high-performing conformer generation and alignment algorithms from biopharmics, and it is backed by rigorous science and benchmarks. A little bit about how this calculation happens in the background. You can start with a 3D structure of an active ligand, and that can come from a variety of places. It can come from a protein ligand crystal structure. It could come from a pharmacophore hypothesis that you may have generated either by comparing a number of known actives with some structural diversity, or it could be simply a low energy conformation of a ligand that you know to be active. Now, when we think about those ligands that we know are actives, we would hold those in a steady static conformation, so as a reference structure, and then we could bring in our list of ideas to populate the conformations of, and then take each of those conformations, align it back to the reference compound, and then get a score. With eSIM 3D, the score is a function of 4 different things. There, of course, is a size component, so a steric component. There's also a coulombic or an electrostatic component, a distribution of the charge around the surfaces of the molecules. But also there's a positional and a directional hydrogen bond acceptor and a positional directional hydrogen bond donor. So coupled together, those factors come together to give a score that really reflects how similar two ligands are. The integration of those factors let us do things like compare new designs to a reference ligand. I've made some changes in my ligand. How does that stack up in that steric, electrostatic, and hydrogen bond acceptor donor layout? I've got some active compounds and I want to get a consensus alignment, so a pharmacophore hypothesis. And now with the release of StarDrop 7.5, we can find new compounds using 3D virtual screening against known actives. So with StarDrop 7.5, we've built on this capability that we released in StarDrop 7.1, and we've brought in the ability to do virtual screening. Now, the capability to screen against a large collection of compounds was in StarDrop 7.1. However, it would be horribly impractical to bring in millions of compounds into StarDrop and try to populate all of the conformations and do the alignment. To this end, what we've done is we've pre-populated the conformations of a variety of commercially available collections that you can download and use freely from our community website. And with that sitting in the background, we can point StarDrop to that file that already has the conformations to do a virtual screen. What's really cool about this is that it is absolutely lightning fast. So if you look at the table that has a little bit of our internal benchmarking on a computer that's very much like mine, which is a 4-core laptop that's just about 3 years old now, I can screen through the enamine stock collection, about 3 million compounds, in under an hour. I'm routinely doing virtual screenings over the lunch hour just to see what comes back. The e-molecules screening collection of about 10 million compounds takes about an afternoon. When the results come back, we get a collection of the top X% where you decide how many results you want to look through at the end, the X% of the best screening compounds, which you can go through and you can compare and contrast both in the alignments together, but also side by side with the surfaces that you see rendered on the structure below that Ed will tell you a little bit about during the live demo. So with this, I am going to turn the microphone and the camera back to Ed for the next stage.
So I've really just got sort of a brief sort of final slide to show, just highlighting one or two other additional enhancements that we have within StarDrop 7.5. These are more about, I put them under the banner of quality of life. So just using StarDrop easier and slicker for you. One of these is making it far easier to generate structural alignments, or rather to align compounds within your data set so they're neatly aligned for exploring SAR. So we can now define a very generalized substructure and then perform a rigid alignment of the compound you bring in. StarDrop wants to honor all the coordinates of the compounds as they're laid out as you bring them in. But using this approach, they'll be sort of flipped and rotated appropriately so that they're lined up, allowing you to sort of easily see differences between the compounds. We've also made it easy to edit structures in place. So if you do bring in a sort of set of compounds and there happens to be a correction that needs to be made, sort of in this particular example, just a subtle change to the stereochemistry, simply double click on a structure, open the editor, and it will replace the compound in your data set for you. And this is just alongside a number of other sort of quality of life improvements we have, such as zooming in on the table using the mouse wheel. Simple things, but improving the overall user experience. So what I'd like to do now is jump to a live demonstration. So what I'm going to do is open up StarDrop and actually show you in practice some of the things that we've just been describing. So here we have StarDrop, and I'm not going to attempt to talk you through all of the core features and the modules as I was sort of describing in my original introduction. I'm really just going to focus on the new features that, particularly those that Ray and Mario have just described. And I'm going to start with virtual screening. And where you can see I've got here on the left-hand side, in the sort of the 3D area of StarDrop, a PDB file I've opened. This is the PDB file to XJX, where I can see this is analysis PIB, which is sort of the co-crystal structure in heat shock protein 90. So I'm going to use this as a starting point for a virtual screen. And just a couple of things that we can see here. Obviously, we can see that we've got sort of this nice pocket. That's why I put the surface on the protein, just so we can see that really the carbonyl and the two hydroxyl groups sort of sit nicely within the pocket here, along with the isopropyl. Whereas we have the pyridine ring sort of sitting out here, which is more sort of solvent exposed. So this is within sort of StarDrop's 3D area, where we have the Surflex eSIM 3D functionality that Ray sort of described very briefly. And again, I'm not going to talk through the various functionality for generating binding hypotheses, alignment similarities, and so on. But I'm going to use this new option that we have here for running a virtual screen. So the one prerequisite of running one of these virtual screens is that you visit our website and download one of the libraries against which you'd like to run that screen. Having done that, we can then choose a virtual screening reference. So in this case, I'll tell it that I want to use 2xJX, and this will grab the co-crystallized structure. It's given me a suggestion for an alternative protonation state. I'm going to accept that. You can see I've got exactly the same compound here in my sort of preview window, but I'm going to use this as my reference when I do my screening. Having chosen my screening reference, I then have one or two options that I can choose. The first of those is to confirm the virtual screening database that I want to use. It will show you the last one you happen to use. In the last case, I happen to run one of the example sets we put there, which is just a random subset of the full library. The full libraries are quite big, so sometimes it's handy to be able to run a screen against a small subset just to get a feel for the functionality. We can choose a parameter scheme, so we can choose, by default, it will give us the highest speed and some good accuracy. But if you do find some interesting results and want to run sort of a further screen again, where you have improved accuracy, which is just a little bit slow, but still very highly performant, we can choose that instead. And we have a couple of options relating to setting the relative volume ratio of the hits we're going to get back compared to the reference, and also the proportion of the top compounds that we'd like to see reported. Remember, the libraries, the smallest of them is sort of getting on for 3 million compounds. So even at the default of 0.1%, I'm looking to get back nearly 3,000 examples. Obviously, we want to up that to 1%, we can do, but we've sort of put a limit there because we're talking about many tens of thousand compounds, particularly for the larger libraries available. Now, the one thing I'm not going to do, and I'm apologizing a couple of times through this live demo, is that I'm not going to do the screen live. As Ray said, it only takes about 45 minutes to run on the full enamine set. But rather than run it on a tiny example, what I already have here in my data set are the results, those 2,700 results that I got from running the full enamine collection against this particular reference here. And we can see that the top result with the highest similarity is this compound here. So if I select this, we can see that superimposed. What I'll actually do to enable us to explore these results a bit is change my display slightly, open up the display options, and what I'm going to do is remove the protein from the display so that we can more easily sort of compare differences. And of course, this is an interesting example. It's the most similarly aligned compound, but we can see it has differences across the compound. We've changed the scaffold, but also in terms of what's potentially sitting within that interesting pocket is quite different. Really, it's only this sort of presence of this carbonyl, which we can see is the same throughout. So this is quite an interesting example, giving us a lot of different areas of the molecule to explore as we try to understand the SAR. If we want to look at this sort of side by side, we can easily do so as Ray described. So sometimes it's handy to be able to sort of make easier comparisons rather than having everything overlaid. But if I move down to, say, this example, the third one down, we can see another example here where there are some much stronger similarities, particularly in terms of this region which was sitting within the pocket. We're obviously missing one of the hydroxyl groups here, but otherwise it's very much the same, but it's very different in terms of the rest of the compound. And maybe we might want to use some of eSIM's functionality for looking at ways we can sort of highlight some of those differences. So if I choose to show a surface, we can either show similarities or we can show differences as a way of picking out different characteristics that could be interesting. And of course, the removal of this hydroxyl group accounts for some of the differences we're expecting to see down here. Just one more example to look at is this example in row number 5. Again, we can see that really this has very strong similarity. It's got the same pyridine ring up at the top here, so there's very strong overlay and alignment up here, but very different in terms of the structure that would potentially be sitting down sort of towards this pocket and interacting with the protein. So it gives us quite a lot of different interesting SAR to explore and to learn about with some of these hits. So obviously we can use the functionality of eSIM 3D to explore the hits that come back. But as Ray said, the hits come back very, very quickly, giving you plenty of opportunity to explore which compounds you think might be interesting. And the IDs that are presented here are those that come from those databases. So if you decide that actually these compounds we're looking at are interesting, we can easily copy these out of StarDrop. We could order up the compounds very easily from whichever compound collection that you have run the screen against. But hopefully that gives you a feel for how seamlessly integrated this new virtual screening functionality is and how it sits neatly within the eSIM 3D module. What I'm going to do now is switch and have a look at some metabolism results instead. So I'm going to switch to the metabolism area and open up another database. And as I said, I was going to do a little bit of apologizing here because, again, the P50 models we have can run in a couple of minutes per molecule. We now have the ability to run enzyme models across a much wider range of enzymes. We can also generate sort of metabolic pathways where we look at multiple generations, where we can consider phase 1 and phase 2 metabolism sort of synchronously. So potentially these models can take a lot longer to run. And so I pre-calculated some results for some compounds just to be able to give you a flavor of the kind of things that come back. So here I've got codeine selected. And as Mario highlighted, we have a few things going on. So firstly, we have our witch enzyme model, which is giving us a steer as towards the fact that we suspect that it's UGTs and salts or P450, which are going to be responsible for metabolism of this compound. And alongside this, in terms of the P450 metabolism, we're anticipating it being 3A4 and 2D6, which are the major P450 isoforms responsible. And this is really reflected in this sort of regioselectivity map that we show up here, where we can see the molecule annotated, highlighting the sites of metabolism we expect to see. So we can see the 2 sites where we anticipate glucuronidation taking place as a result of UGT. And if I hover over those sites, we can see the expected metabolite. And equally, we can see the sites where it's anticipating the demethylation to take place as a result of 3A4 metabolism, again, here and up here, and this site up here is also where we would anticipate all 2d6 metabolism taking place. Mario also highlighted the fact that we have our metabolic landscape that we can look at, which gives us an idea of the lability of the compound in terms of its ease with which P450 metabolism can occur, particularly in terms of 3A4 metabolism. In this case, we're highlighting the fact there are two highly labile sites. So if I hover over those sites, we can see them picked out on the reference compound above, along with the annotations. So as Mario highlighted, if we were making modifications to this compound to try and improve its stability, we probably have to make changes at both sites. If we only made one of them more stable, we'd probably see metabolic switching going on, and we might not expect to see much of a change in the overall rate of metabolism. So as well as being able to see the metabolites as we hover over the individual annotations, we can pull up a table. I changed this here, so we can pull up a table which gives us all the details of the metabolites we'd expect to see. And if we want to, we can export that full table of metabolites out into its own data set if we wanted to run calculations on them to explore potential toxicities or look at risks or opportunities associated with those metabolites themselves. Another possibility we have for looking at metabolites comes from our ability to generate metabolic pathways. So one of the things you'll notice up here is that we've only calculated predictions for some of the enzymes, P50s, UGTs, and salts, and not for AOX and FMOs. And this is because the Which enzyme model has given us a steer that these are the most likely ones, and these are the ones that it's worth that sort of computational investment in. But we do have a number of options available to us that allow us to choose which of the different models that it is we wish to run. So you don't have to be steered by Which enzyme if you want to pick and choose sort of the specific enzymes on which to focus. We also have the opportunity to tell it not just to generate one generation of metabolites, but actually generate a metabolic pathway. And when we do this, we're able to then pop our results into the kind of schematic that Mario showed using card view, where we see not just that first generation of metabolites, but also those that follow on afterwards. And I've sort of pre-prepared this by coloring it so that you can see those metabolites which are a result of P450 metabolism, as opposed to those which are a result of UGT. As I said, all of the details that these are available within the metabolites view we have down here, where we have sort of that complete table of not just the first generation, but the second generation metabolites. What we're seeing in our view to the right is the most likely ones. So it enables you to sort of either take that sort of view on the most likely or all the gory details, depending upon what you'd like to explore. One last thing that I wanted to mention comes back to what Mario highlighted relating to the preclinical species. So we have an example in this data set, which is alnidamol, where we can see that it's aldehyde oxidase, which is predicted to be the most likely route of metabolism. And we know that guinea pigs and rhesus monkeys display similar AOX activity to humans. So in this case, this would already be a steer that we might want to consider those species in any sort of follow-up sort of animal studies that we carry out. If instead we look at something like sildenafil, where we anticipate sort of P450 metabolism dominating the picture, we can see here that we have a particular site metabolism that we anticipate being a majority of metabolism occurring. If we look at the results from the preclinical model, we can see from that this same site we would anticipate seeing in humans is also a primary site of metabolism in mouse, rat, and dog. So we'd be quite comfortable that perhaps any of these models could give us a steer towards this. If we look at some of the other sites that we see within, or predicting to see within human metabolism, we can see it's really mouse and dog that sort of exhibit these sites as well. So it's probably mouse and dog that we choose as the two models which are sort of most representative of humans if we were sort of following this up in a preclinical study. So these models can sort of allow us to sort of make those decisions about the studies that we want to sort of carry out further. Last thing I wanted to sort of touch on related to some of those sort of quality of life features I mentioned. I sort of described the fact we can easily align compounds. So I've opened up a data set here where I've got buspirone and a number of its analogues, which are all sort of laid out as sort of faithfully based upon the way we imported them into our data set. But we now have the ability, if I were to select buspirone itself, we have the ability to choose to align the 2D diagrams in this set. And we can do this now by defining whatever scaffold we want to represent the way we'd like these laid out. So if I were to remove this region here and make this part here, what I'm going to do is put in a variable linker here to reflect the fact that so this region of the molecule, in some cases, we have a sort of 5-member ring. But we can actually sort of interact with our scaffold, as it were, our scaffold structure, and be as specific about subtleties around bond counts and hydrogen counts or generality of elements as we wish. We can do exactly the same with bonds. So we can define a very general structure to describe the way we want to lay out the compounds, depending upon the set of compounds that you're looking to align. But having defined this, if I hit the align button, we will then see that it's aligned all the compounds in the set because they all match this pattern that I've described, and they're all now laid out faithfully, rotated and flipped appropriately in order to match up with that alignment. I also mentioned the ease with which we can edit compounds now. It's simply a case of double-clicking on a compound, making whatever change. Presumably it's likely to be a small change that you notice where a compound hasn't been necessarily faithfully represented before you imported it. In this case, if I make a very subtle change here and click OK, it'll confirm I really want to replace the molecule in my set, because what StarDrop will then do is look at anything that's been calculated on the basis of that structure and update it, or in this case, where we've got long-running calculations, simply reflect the fact we haven't calculated these yet. So any ADME-QSAR predictions will be updated. In the case of the metabolism results, it doesn't automatically trigger prediction, but it does reflect the fact that we haven't calculated the results for this particular compound. We can, of course, undo that. So I'll just do a quick Control-Z to undo. So it's not a sort of a terminal change we've made, if we didn't want to, but it does make that change very easy to make. And finally, I mentioned sort of things like zooming. If I hold down the Control button, if you want StarDrop data sets to look bigger and have sort of bigger cells all around, it's very much up to you to just zoom into the level that's useful for you to work with. And it's all yours, and StarDrop will remember this and save it the next time you come in and work with it. So hopefully that gives you a flavor for some of the things that we've now introduced in the newest version of StarDrop, and hopefully these are all bits of functionality that you'll be keen to give a go yourself, and we'd love to hear your feedback on that. What I'm going to do is pop back into presentation and really just wrap up with 1 or 2 conclusions. As I mentioned, what we'll do in just a second is come to the question you've been posing, so we can see a number of them coming in. But if any of you do have questions, now's a good time to type them into that box. But hopefully I've given you sort of a feel. I gave you a very quick introduction to StarDrop in terms of it being a complete platform for small molecule design, optimization, and data analysis, and obviously a run through the new metabolism and virtual screening functionality that we've added. There's a huge amount more information on our website, as Catherine described at the beginning, where we not only have detailed information about the different pieces of functionality, but we also have case studies which describe how it's been used in sort of different scenarios, giving you a feel for how the different pieces of functionality can be used together. Our online community, again, has the various previous webinars and videos and tutorials we've put together. So if you particularly want information about specific features of StarDrop, again, or sort of a longer running sort of worked example, we have videos and downloadable examples you can get there, along with publications and presentations and posters, really giving you a lot more depth of information behind the science that sits behind StarDrop. And of course, I touched in my when I talked about customization on the fact there are a number of add-ons for StarDrop. Plenty of those can be downloaded directly from our community site. So if you want to give them a try, please feel free to do so. And of course, you are welcome at any point to contact us for assistance with anything we've talked about today. At this point, I'm going to hand back to Catherine, and we're going to take a look at some of the questions that have been coming in.
Fab, thanks, Ed. And yeah, just a reminder, click that Q&A button. I can see that a lot of you have already asked some questions. That's fantastic. I'm going to kick things off with a question for Mario. In the P450 module, carbon atoms were numbered, but I don't see that in the new metabolism model. How can we identify the atoms from the histogram in this new module?
Yeah, thank you for the question. So yes, the atoms are not numbered per se, but if you open up StarDrop and you use the metabolism module and hover over the specific entries in the histogram, then the atoms which are related to these specific entries in the histogram will pop up, and I believe Ed will demonstrate that right away. As you can see, the atoms get circled. And we removed the numbers because the numbers were essentially related to the smile string. However, in the graphical user interface, we don't actually display the smile string, so it didn't really make any sense from the user perspective.
That's great. Thank you so much, Mario. Our next question is for Ray on the virtual screening side. So how does this work for kinases or other cases where there are explicit hydrogen bond interactions that should be exploited to achieve binding?
Oh, that's a good question. So in the scoring function for the Surflex eSIM 3D alignment, there are those components that go into it. So there's a volume, so a surface overlap. There's also the distribution of electrostatics, but there are explicit terms for both positional and directional hydrogen bond acceptors and hydrogen bond donors. So what comes out at the end are hits that get the right collection of hydrogen bond donor and acceptor motifs without any fiddling or messing about with the scoring function.
Great, thanks, Ray. Next question is for Mario again. Which preclinical species models do we have, and why?
So yeah, the preclinical species or the models for preclinical species we have at the moment are for mice, for rats, and for dogs. And the reason we don't have any additional preclinical models is the data. So these three species had the most data in the literature, and for any other species, I think it's just a matter of time where we can start training models for other species as well. We just have to wait for publicly available data for those species. Thank you.
That's great. Thank you very much. Next question is back to Ray, and it's about whether it's possible to run eSIM virtual screening on Enamine's real space.
Oh, that's 6 billion compounds. So I think it's very much of the case of I could walk to Poland if I have lots of time. So my colleagues, my biopharmics colleagues, have done some back of envelope calculations that there would be an initial investment to generate all of the confirmations using Amazon Web Services EC2 instances. And once they are generated, the estimate was that by using 136 core EC2 instances, the enamine collection can be done in a couple of hours. The real space can be done within a couple hours. So, but it's very much well, there's a philosophical discussion around whether we should do it, but it appears that we can do it. So there's no plans at the moment. We've not discussed this internally, whether we would go in this direction, but it is something that is on our radar.
That's great. Thank you very much, Ray. Another question for Mario about whether there are any isoform specific models for sulfur transferases.
Yes, so indeed, the only model in the human-specific models, which is not isoform-specific, is the sulfotransferase models. And unfortunately, we kind of have the same situation as we had with the preclinical species. There is just not enough isoform-specific publicly available data for sulfotransferases. So what we did, we gathered all of the data we could for sulfotransferases and trained a general model. And the reason why it's fairly good is because it is our assumption that in practice, only one or maybe two relevant isoforms do most of the drug-like compound metabolism for sulfotransferases. So I guess the answer is very similar to the previous question. We just need to wait for more published data for sulfotransferases. I should say more isoform-specific data. Thank you.
That's great. Thank you, Mario. Just looking at the questions, I think we've got another one for Mario. So if you don't have experimental data, then StarDrop could lead you to optimize something that doesn't need to be optimized. Is that true? I'm assuming in relation to the metabolism module.
So yes and no. I think the problem might arise when you just use one enzyme or models for one enzyme family. In this case, yes, that could be the case. But if you go through the kind of the steps that first you kind of check the which enzyme model, which tells you which which enzyme family is most likely to metabolize your compound, and then continue with the specific enzyme family, then it shouldn't be a problem. Or when you go through the metabolic pathway calculations, then it shouldn't be a problem as well. However, if your compound gets metabolized by, say, UGTs, but you only study the P450 metabolism, then yes, this could lead to a situation where you start optimizing your compound, when in reality, you shouldn't optimize your compound for the P450 metabolism. So it's a yes and no situation.
Great, thank you very much, Mario. Okay, I think the next question is just a final question actually for Ed, just to remind everyone how they can access StarDrop 7.5. What should they do if they want to upgrade to StarDrop 7.5?
You've saved a nice easy one for me. So if you're already using StarDrop, then you should have heard from us. We've been in touch with everybody. So you or your colleagues should have received some information about how you can get hold of StarDrop. But if you haven't, please send us a message to info@optibrium.com or support@optibrium.com, because we'll be more than happy to help you get hold of it. It's available to everyone who licenses StarDrop, already has access to the latest version of it. So please just let us know that you're interested. If you're not already using StarDrop, you're also welcome to use exactly the same methods, because you're very welcome to give it a try. So please get in touch in either scenario if there's anything we can do to assist you with.
That's great. Thank you very much, Ed. So we've reached the end of today's webinar. So thanks very much, everyone, for all of your questions. And as Ed says, if you have anything that you want to still discuss, if you have any questions or you're interested in using StarDrop 7.5, just get in touch via email to the info@optibrium.com email address, which is now up on the screen. So all that's left for me to do today is to thank all of our speakers and thank you all for attending. Hope you enjoyed today's webinar, and we'll see you soon.